transfer of amino group from one amino acid to a keto acid to form another non-essential amino acid and a keto acid. The next step was deamination. In case of transamination, most common amino acid that was formed was glutamate. এরা কিছু কিছু তাও পড়েছে, এই এদিকে লোকজন কিছু পড়েনি. So glutamate was the end product of transamination reaction. In case of deamination, what happened? Glutamate was converted into ammonium ion and alpha ketoglutarate by oxidative deamination reaction, and it was mediated by the enzyme glutamate dehydrogenase, and it was exclusive in liver cells, liver mitochondria. Okay, and the other steps were transport of ammonia, where glutamate was combining with ammonia to form glutamine, and that glutamine was further carried to liver, right? From all other cells, all other tissues, and from muscles. What was the amino acid that carries the amino group? It is alanine. Okay, let's look for today's specific learning object. So we will learn about urea cycle today, as urea is the end product or excretory product of protein metabolism in all form. All nitrogenous compounds are excreted as urea. So we should know about few salient features of urea and its importance, and then the outline of urea cycle, its steps and major important enzymes, and the significance of urea cycle along with its regulation. Okay, clear? So here is a quick recap of last day's class. What is transamination? Transfer of amino group from one amino acid to a keto acid in presence of transaminase enzyme, and the cofactor was what? Pyridoxal phosphate. Next, it is deamination. What is deamination? Removal of the amino group in form of ammonium ion, and it can be oxidative or it can be non-oxidative. Oxidative deamination is mediated by glutamate dehydrogenase enzyme. Okay, now transport of ammonia. What happens? The free ammonium is attached with glutamate to form glutamine, and that glutamine again divides into glutamate and ammonium ion by glutaminase enzyme. Okay, so this is the transport form of ammonia. And the last part is urea cycle. What happens? The free ammonium group, they are incorporated in our liver and forms urea. Okay, so why urea is excreted? The structure of urea. Okay, look, here are two nitrogen. Okay, two nitrogens are there along with a carbon central molecule. Now one nitrogen of urea comes from the ammonium group. Okay, that was getting liberated in the liver, and the second nitrogen group is from aspartate. Okay, so we will see later on in the urea cycle itself how aspartate gives the second nitrogen group, but this is an important MCQ question also, that what are the sources of two nitrogen groups of urea. One is from ammonium ion, another is from aspartate, and the carbon is from carbon dioxide that is produced in our PCA cycle. Okay, it is the bicarbonate ion. So carbon dioxide with H2O, it forms that bicarbonate form, and it is incorporated to form the urea. Okay, clear? Bojha geche? So what are the source or the component source of urea if asked? Is it clear? The carbon is from carbon dioxide, from TCA cycle, one nitrogen is from ammonia and another is from aspartate. Okay. So here are the few salient features of urea. First of all, we should know the reference range or the normal range in humans, irrespective of age and sex and body weight. Okay. So this is the normal reference range, or reference range, what is it? 15 to 40 milligram per dl. 15 to 40 milligram per dl is the reference range of urea. Urea is synthesized in liver. You all know urea cycle occurs only in liver, and we will know why. We will learn why. Okay. So it is the end product of protein metabolism. All these are very easy points as it seems now, but in your exam if asked, you always fumble. Okay. So and most important thing about urea is it is an indicator of renal function test. So can you name another indicator for renal function test along with urea? That is creatinine. Very good. So urea, creatinine or together they forms the renal function test parameters. Okay. And now it accounts for 80 to 90% of nitrogen containing substances in urine. I told you the major excretory product of nitrogen metabolism or protein metabolism is by urea, excreted by urea. Okay. And urea is a normal constituent of urine, not abnormal. All of you have learned many other abnormal constituents in your practical, but urea is a normal constituent of urine. Okay. Next, this is just for a rough estimate knowledge that how much urea we excrete from our body. And here another question may be asked, why urea is the ideal nitrogenous excretory product? So because it is highly water soluble. First of all, urea is highly water soluble and it can be diffused through the tissues, and secondly it is non-toxic and metabolically inert compound. Okay. So it is non-toxic. Ammonia was toxic. Do you remember from your last class why ammonia was toxic? Why accumulation of ammonia would lead to toxicity? Anyone? As increased ammonia will lead to formation of glutamate by combining with alpha ketoglutarate. Alpha ketoglutarate is a intermediate product of TCA cycle. So TCA cycle will be hampered. So exactly, so less ATP production will be there. TCA cycle if hampered, it will produce less ATP. Second, it will increase the production of glutamine. It will help in accumulation of water in brain. So there will be edema in brain. Next of all, it will lead to decreased amount of glutamate in brain. Increased amount of glutamine will lead to decreased amount of glutamate in brain, and that's why glutamate was producing GABA, which was an inhibitory neurotransmitter. So GABA will be decreased. So all these things will lead to neurological symptoms like seizure, tremor, slurring of speech and blurring of vision. Okay. So all these things. Now coming to urea cycle proper. So now we all know about urea a little bit. Okay. So this cycle is the first metabolic cycle discovered. Okay. Even before TCA cycle, it was discovered in 1932 by Hans Krebs and Kurt Henslett. So this cycle is also known as Krebs-Henslett cycle. Okay. And ornithine cycle, this is also known as ornithine cycle because ornithine is an intermediate of this cycle, and it is also very important. Okay. So what is the site of urea cycle? Liver, and there are 5 steps and 5 enzymes. Okay. So the first 2 steps occur in mitochondria and last 3 steps occur in cytosol. So this is just to remember, and this is a schematic representation of a cell, liver cell, where the green outline is a cell and the inner part is denoted as mitochondria. So, for your easy remember here, in mitochondria first two steps are they take place. One is synthesis of, first step is synthesis of carbamoyl phosphate. Okay. First step is synthesis of carbamoyl phosphate and the enzyme is carbamoyl phosphate synthetase 1. The second step is formation of citrulline and the enzyme is ornithine transcarbamoylase. Okay. Next three steps. Third step is synthesis of arginosuccinate by the enzyme arginosuccinate synthetase. Fourth is formation of arginine and that is by arginosuccinase. Okay. Fifth is formation of ornithine and urea by the enzyme arginase. Clear? These 5 steps and 5 enzymes. Okay. Now we will see the urea cycle itself. The first step, it is synthesis of carbamoyl phosphate in mitochondria. So, I told you the first 2 steps occur in mitochondria. So, and from the structure of urea I told you the one nitrogen is from ammonia and the carbon is from carbon dioxide. So, look, here is the carbon dioxide, okay, and it forms HCO3 and combines with the ammonium ion to form the carbamoyl phosphate. Okay. So, carbamoyl phosphate is formed from carbon dioxide and ammonia along with H2O. Okay. And this is energy taking step. Here 2 ATPs are used and 2 ADP and inorganic phosphates are released. Okay. The main enzyme here is carbamoyl phosphate synthetase 1. This is the rate limiting step and regulatory step of this overall urea cycle, and this reaction, this is allosterically activated by N-acetylglutamate. Okay. N-acetylglutamate is nothing but acetyl-CoA. When reacts with glutamate, it forms N-acetylglutamate. Okay. Clear? This step is clear. Carbon dioxide with ammonia with water forms carbamoyl phosphate in presence of the enzyme carbamoyl phosphate synthetase 1, and it is allosterically regulated by NAG, that is N-acetylglutamate. This is the irreversible rate limiting step. Now the second step, what happens? This carbamoyl group is transferred to ornithine to form citrulline. Okay. So, why this cycle was called ornithine cycle? Now you will understand. Okay. The second step, in the second step, ornithine is required to take this carbamoyl group, okay, from the carbamoyl phosphate compound, and it will form citrulline and one inorganic phosphate will be released. Okay. So, the whole compound will be transferred to ornithine and one inorganic phosphate will be released and citrulline will be formed. This is also occurring in mitochondria. Clear? And here the enzyme is ornithine transcarbamoylase. Okay. Now you can see that ornithine is coming from cytosol to mitochondria. Here is the trick. Ornithine lies in the cytosol. Ornithine is in the cytosol. So it requires to come inside the mitochondria. Here there is a transporter, ornithine citrulline transporter. It transports one molecule of ornithine inside the mitochondria and it takes back one citrulline to the cytosol. Okay. So here is a transporter, ornithine transporter or ornithine citrulline transporter. So the second step that also occurs in mitochondria is facilitated by the enzyme ornithine transcarbamoylase or OTC. Okay, clear? After forming citrulline, the citrulline will come outside the mitochondria into the cytosol. by the same transporter, ornithine citrulline transporter. And in cytosol, all other steps will occur. Okay, first two steps that occurred in mitochondria and all other three steps will occur in cytosol. So what happens in cytosol, the citrulline that comes out will join with aspartate. The aspartate which will give the second nitrogen of urea. Remember from the structure of urea, the second nitrogen was from aspartate. So aspartate is another amino acid, and where we are getting the aspartate from transamination reaction. Can you name one transamination reaction? And the enzyme required for production of aspartate? AST, aspartate transaminase, and that was formed from oxaloacetate. Oxaloacetate with glutamate will form alpha ketoglutarate and aspartate. Okay. So aspartate will join with citrulline to form arginosuccinate. Arginosuccinate, and here again one molecule of ATP is getting used, but see, the end product of ATP is AMP and a pyrophosphate, inorganic pyrophosphate, which will further be broken into two inorganic phosphates. So this is equivalent to 2 ATPs. Okay, as two high energy bonds are broken here. One ATP is getting used, but the bonds that are energy bonds are being broken is in number 2, because 2 inorganic phosphates are getting released. So up to this 2 ATP, 2 ATPs were used here, and 1 ATP equivalent to 2 ATPs. Is it clear? Don't confuse that here only 1 ATP is getting used, so why ma'am you are telling ki 2 ATPs are being used? Because 2 inorganic phosphates are being released at the end. So 2 high energy bonds are used here, that is equivalent to 2 ATPs. Okay, and here the enzyme is arginosuccinate synthetase. Okay, so this is the third step where citrulline combines with aspartate and forms arginosuccinate. Clear? E porjonto clear? Fourth step. What happens in fourth step? Arginosuccinate by arginosuccinase or arginosuccinate lyase is getting converted into arginine, and fumarate is released. Okay, so fumarate is getting released here, and the cycle goes with arginine. This step is very easy, arginosuccinase or arginosuccinate lyase. Incorporate with arginine, and by the enzyme arginase, urea will be released and arginine will get converted into ornithine. So this ornithine is what was in the cytosol. Okay, and it will again get incorporated into the urea cycle. বোঝা গেছে? Clear এইটুকু? So arginine যেটা তৈরি হলো, arginine will be hydrolyzed by the arginase enzyme to form urea and ornithine. Okay, so why this cycle, urea cycle, is also known as ornithine cycle? Is it clear now? So from the second step to fifth step, ornithine is involved. Without ornithine, citrulline will not be formed, and without forming citrulline, here urea will not get released. Okay, clear? So now coming to the fate of the urea produced. Fate of urea, what will happen to the urea that is being produced in urea cycle? So the urea will be excreted by kidneys in urine, right? I told you urea is highly water soluble, so it will get dissolved in blood and it will be eliminated in our urine. And a part of urea is also transferred to intestines, okay, by blood circulation and get released in our feces also. Now see, this enzyme arginase, the fifth step enzyme, this is exclusive for urea production. Why? Because this enzyme is only present in liver, not in any other organs, okay. So up to this, up to arginine synthesis, this might occur in some other organs also, but the last step, that is arginine to ornithine production and urea release, will not take place anywhere other than liver. Now coming to the significance or importance of urea cycle. One, you know, that is production of urea. Another thing is the fumarate that was produced in the as a byproduct of urea cycle, that will be incorporated in TCA cycle or citric acid cycle. Clear? How? So here fumarate will be converted to malate and malate will be converted to oxaloacetate and thus the TCA cycle will run. And this fumarate to malate, bifumarase enzyme and malate from oxaloacetate by malate dehydrogenase, all this occurs inside mitochondria. The TCA cycle occurs in mitochondria. So clear, this fumarate will go back to mitochondria and this will, the TCA cycle will run. So this fumarate links urea cycle to TCA cycle. Is it clear? So here are two cycles going side by side. That's why urea cycle with TCA cycle is known as Krebs bicycle or urea bicycle. Okay, and biochemically these cycles are very important because one is linked with amino acid metabolism and another is the energy production pathway. So is it clear how any amino acid metabolism will lead to energy production other than their ketogenic or glucogenic fate? By urea cycle also fumarate is getting produced and fumarate will go to the TCA cycle. And now see here, so TCA cycle has been taught in your class, okay, right? So can you tell me from malate to oxaloacetate how this NADH is produced, and NADH is equivalent to how much ATPs? 2.5. So if we see the whole energetics of this cycle, okay, another thing, here the oxaloacetate that was being produced from malate can also produce aspartate. As I told you, aspartate from transamination. I told you aspartate was the end product of transamination from glutamate and oxaloacetate reaction, AST, aspartate transaminase reaction. So oxaloacetate will further can form aspartate by glutamate and AST. Okay, and gluconeogenesis can also occur from oxaloacetate. Now the energetics. So 4 ATPs were used in urea cycle, right? And in TCA cycle, fumarate, one molecule of fumarate will give rise to 2.5 ATPs, like from one NADH, right? One NADH will be produced because fumarate will be converted into malate and malate will get converted into oxaloacetate. So this NADH is equivalent to 2.5 ATPs. So the net ATP consumed will be 4 minus 2.5, that is 1.5 ATP. Clear to all? This is also asked in your exam, that write the energy breakdown of urea cycle. Is it clear? Here 2 ATPs were utilized in the first step and in the third step 1 ATP but equivalent to 2 ATPs were used. Clear? And from malate to oxaloacetate 1 NADH will be produced, and that is equivalent to 2.5 ATPs. So the net ATP utilization is 1.5. Clear? Clear to all? Okay. Now coming to the significance of urea cycle. You all should answer by now. The main aim of urea cycle is to detoxify the ammonia that is getting produced in our body and its excretion. And along with ammonia, it also excretes the carbon dioxide produced. Right? So, two waste products are disposed. One is ammonia, another is carbon dioxide produced in our tissue. Third is regulation of blood pH. How? Because the more ammonia is getting produced and the more urea cycle is running, HCO3 is also being utilized. So, HCO3 or bicarbonate, that is the alkaline component of our blood. Okay. So, alkaline component, if the urea cycle is hampered anyway, the alkaline component will rise and it will alter the acid-base balance. Fourth is biosynthesis of arginine. Why arginine is important? Because we will study in our glycine metabolism also that arginine is important for creatine synthesis. Okay. So, creatine is a important product for our muscle, and that is why arginine to creatine production is also important. And also the ornithine production is going on in urea cycle, and ornithine is important precursor for polyamines. These are putrescine, spermidine, spermine, these are polyamines. Okay. Now come to the urea cycle regulation. I told you about the main enzyme and the regulatory step. What was it? Which step of urea cycle was the regulatory step? First step, or the formation of carbamoyl phosphate by the enzyme carbamoyl phosphate synthetase 1. Right? And it was allosterically regulated by which compound? N-acetylglutamate. Here glutamate and acetyl-CoA will form N-acetylglutamate by the enzyme acetylglutamate synthase. Okay, acetylglutamate synthase will help to form the N-acetylglutamate compound, which will bind with carbamoyl phosphate synthetase because it is allosteric activator. Okay, so allosterically it will promote this reaction, the formation of carbamoyl phosphate. And what are the factors that will lead to formation of more N-acetylglutamate? That is high protein diet, arginine and starvation. In case of starvation, what happens? Protein breakdown increases. So, increased protein in our blood will lead to increased production of N-acetylglutamate, and thus this reaction will move forward. And this is one way, single directional, irreversible reaction, rate limiting. Now, the CPS1 or carbamoyl phosphate synthetase 1 is seen in urea cycle, and the similar enzyme, that is CPS2 carbamoyl phosphate synthetase 2, it is seen in pyrimidine synthesis. Okay. So, the difference between CPS1 and CPS2 is often asked. So, you may write these points. CPS1 is located in mitochondria, as the first step was occurring in mitochondria in urea cycle, but in case of CPS2, this enzyme is present in cytosol. Here CPS1 requires a cofactor, that is NAG, but in case of CPS2, no cofactor required. In case of CPS1, the nitrogen donor was ammonia, but in case of CPS2, it will be glutamine, and here there is no inhibitor, but here CTP, cytidine triphosphate, is the inhibitor in case of CPS2. Clear? Is this clear for everyone? CPS1 and CPS2. Okay, now coming to the MCQs. Before going into the MCQs, just for a quick revision of urea cycle, tell me the another name of urea cycle, ornithine cycle, and it was discovered by Krebs and Henslett. So, also named as Krebs-Henslett cycle. Okay, and what is the site of urea cycle? It occurs in liver because the enzyme arginase is only present in liver cells. Okay, now coming to the steps. How many steps are there in urea cycle? 5 steps and 5 major enzymes are there. So, step number 1 occurs in mitochondria, and that is incorporation of carbon dioxide and ammonium ion with water to form carbamoyl phosphate by the enzyme carbamoyl phosphate synthetase 1, and the allosteric activator here is Nag. Okay, and this step requires how many ATPs? 2 ATPs. 2 ATPs are required, and 2 ADP and 2 inorganic phosphate is released. Next step is transfer of carbamoyl group to ornithine to form citrulline, and this is facilitated by the enzyme ornithine carbamoyl transcarbamoylase. Okay, and this ornithine comes from cytosol by the transporter protein ornithine transporter, and this transporter also helps in the moving out of citrulline to the cytosol. The citrulline that was being produced in the mitochondria will come out into the cytosol. Now, in cytosol, the step number 3, 4 and 5 will occur. So, what is the step 3? Citrulline will join the aspartate to form arginosuccinate, and the enzyme is synthetase. Okay, and this aspartate is coming from transamination, and the source is oxaloacetate. Okay, and next step, step 4 is, and the third step was using how many ATPs? 1 ATP, but two equivalent high energy bonds. Okay, and the fourth step is arginine, and the enzyme is lyase. Okay, and what is the byproduct here? Fumarate. So, fumarate is the link between urea cycle and TCA cycle. This will be converted into malate and oxaloacetate subsequently, and this will go into the proper TCA cycle. Okay, from arginine, what will happen? The last step or the fifth step, arginine to ornithine will be formed and the urea will get released, and this is a hydrolysis process. Okay, water will be, one molecule of water will be incorporated, and what is the enzyme? Arginase. Very good. So, what is the fate of urea that is just produced? So, it is mainly excreted in the urine, and some amount may be excreted in the feces also. Okay, why the reaction, the urea cycle is exclusive in liver? Because of the enzyme arginase. Arginase is not present in kidney or intestine, whereas up to arginine, all the reactions they can occur in other organs also, but the last step or the final step, that is exclusive for liver. Okay, and what are the energy breakdown of urea cycle? So, total 4 high energy bonds were required and 1 NADH in TCA cycle will be formed. So, 4 minus 2.5, that is 1.5 ATP altogether is the net utilization. Okay, and what are the other outcomes for fumarate? Like fumarate will be converted into oxaloacetate, and that oxaloacetate can go into TCA cycle, or it can be converted to aspartate, or it can be used in gluconeogenesis. Okay, so these are the overall urea cycle. And what is the regulation of urea cycle? Regulation of urea cycle was by the first step enzyme, that is CPS1, and it was allosterically regulated by Nag, and Nag is regulated by high protein diet starvation, that is more protein breakdown or presence of arginine, more increased concentration of arginine. So, is it up to this clear for everyone? Okay, now come to the MCQs. Urea is synthesized in liver. Regulatory enzyme of urea cycle is CPS1. Okay. NAG is allosteric activator of CPS1. এখন তো very easy. আচ্ছা, তারপর the 2 nitrogen atoms of urea are contributed by, বল পেছন থেকে বল, you. What is the answer? Option C, ammonia and aspartate. Good. Okay. Number of high energy phosphates used in the formation of one molecule of urea is 4. Okay, amino acid that is continuously regenerated and recycled in urea cycle is ornithine. Okay, enzyme of urea cycle exclusively present in liver is arginase. Metabolite released in urea cycle that enters TCA cycle is fumarate. Easy হয়ে গেছে তাই না কোয়েশ্চেন গুলো? আচ্ছা, amino acid of urea cycle that serves as precursor of polyamines. Ornithine. Okay, arginine is a precursor of creatine. Okay, the last MCQ, that is metabolite of TCA cycle on transamination that produces an amino acid that takes part in urea cycle.